Usually when we talk about, medical diagnosis, there's this expectation of clinical precision. Like it feels almost engineering adjacent.
Right, very cut and dry.
Exactly. Somebody breaks their arm, the x-ray shows that jagged white line on the radius, and the doctor just points to the screen and says, there is the fracture. It's binary. It's clean.
Yeah, we definitely crave that visual confirmation. I mean, we like biological systems to be neatly categorized and, well, immediately visible to the naked eye. It gives us this sense of diagnostic security.
But then, you step into the world of immunohematology, and suddenly that x-ray machine is completely useless.
Completely useless.
We're looking at a diagnostic landscape that is entirely invisible. It's operating on this microscopic molecular level inside a patient's veins. I mean, we are dealing with immune wars, shifting antigen expressions, and just incredibly subtle chemical reactions.
It is basically the definition of diagnostic muddy waters. The physical elements we're testing, like the glycoproteins, the glycolipids, the immunoglobulins, we can't actually see them.
Right, you're just looking at the aftermath.
Exactly. We only observe their secondary effects. So we're forced to deduce the invisible architecture of a patient's immune system based entirely on how their blood behaves in a test tube.
Yeah.
Under very specific manipulated conditions.
Which brings us to our mission for today. We are speaking directly to you, the listener, because we know you are in the trenches right now. You are prepping for your ASCP MLS board exam, and, well, you're staring down the immunohematology section.
Yeah, and that section is a notorious hurdle for medical laboratory scientists. It demands way more than just, rote memorization of charts.
Oh, definitely. Memorization is not going to save you here.
No, it really won't. I mean, it'll only get you so far when a board question presents this complex, multi-layered patient discrepancy. Passing requires an actual functional, dynamic understanding of how these biological systems interact. You really need to know the why behind the what.
So, to get you there, we are treating this session as your ultimate auditory study guide. Our source material is this dense stack of blood bank practice questions, but, and this is key, we are not just going to read questions and recite the answers.
Right, because that's passive and honestly pretty ineffective.
So ineffective. Instead, we are executing a focused study plan. We are going full phase 1 concept extraction.
Our main objective today is to extract the core mechanisms, the underlying genetic rules, and the immunological principles behind every single concept tested in your material. And we'll definitely be flagging the absolute high-yield topics that have a really high statistical probability of showing up on your exam.
We're also going to track the trouble spots, the lookalike distractors and the clinical traps that exam writers absolutely love to set.
They do love their traps.
They really do. So we're going to flow logically through the foundational categories. First, we'll establish the raw blueprint, the genetics and immunology of blood groups.
And then we'll take those rules and apply them to the big two, which are the ABO and RH systems.
Right. And from there, we transition to the actual bench work. We'll walk through the detective work of antibody testing and identification, followed by the incredibly strict matchmaking rules of compatibility testing.
And finally, we will explore the clinical fallout when these systems clash, so transfusion reactions and hemolytic disease of the fetus and newborn, or HDFN.
So if you're feeling overwhelmed by the sheer volume of phenotypes, enhancement media, and bizarre antigen names, just take a breath. By the end of this deep dive, the underlying logic is going to actually make sense.
You won't just have flashcards in your head. You will actually understand the biochemical machinery.
Let's start with the absolute foundation. I mean, before we can even think about matching blood for a trauma patient, we have to understand the genetic rules of how antigens are actually inherited.
Yeah, the best place to start there is differentiating what we test in the lab versus what actually exists in the patient's DNA. So a fundamental distinction that constantly shows up on exams is the difference between phenotype and genotype.
This is huge.
It is. When you, as a technologist, put a drop of patient blood in a tube, add a reagent anti-sera, and you observe for agglutination, you're looking exclusively at the phenotype.
Right. You're observing the physical, structural expression of the antigen, like the actual protein or sugar that successfully made it to the surface of the red blood cell.
Exactly. But that tube reaction tells you very little about the complete genetic code that's hidden in the nucleus of the patient's cells.
Take the ABO system as a primary example of this. An individual inherits one ABO gene from their mother and one from their father. Let's say they inherit an A gene and an O gene. Their genetic makeup, their genotype is AO.
However, the O gene is what we call an amorph.
An amorph meaning it doesn't actually code for a functional enzyme that builds an antigen. It's essentially just a genetic blank space in terms of physical expression, right?
Perfectly stated. Therefore, the red blood cell membrane will only display the structures built by the functional A gene. So when you test this patient's blood in the lab, it will violently agglutinate with an anti-A reagent. Their serological expression, their phenotype is simply group A.
And unless you perform molecular genotyping using PCR techniques, which, let's be real, is way beyond routine bench testing, you cannot serologically distinguish an individual who is genotype AO from an individual who is genotype AA.
You really can't. They both phenotype exactly the same. They both look like group A on the bench.
And that distinction is critical because the exam will constantly try to trick you by, mixing phenotypic serology results with genetic family tree questions. They love doing that.
Oh, absolutely.
But let's look at a scenario where the underlying genetics do drastically alter our test tube results. I am throwing a massive high-yield flag on this next concept. Let's talk about the dosage effect.
Oh, dosage. This is a favorite concept for exam writers because it bridges genetics directly to observable lab phenomena. To understand dosage, you really have to visualize the surface area of a red blood cell.
It's limited real estate.
Exactly. It's prime, limited real estate. Let's use the KEL system as an example here. An individual has a certain number of binding sites on their cell membrane designated specifically for the KEL glycoprotein.
Right. So if an individual inherits the exact same allele from both parents, say the gene for the big K antigen, they are genetically homozygous.
And because both of those genes are actively pumping out the big K antigen, every single available piece of KEL real estate on that red cell membrane is occupied by a big K antigen. We call this a double dose.
The membrane is just saturated with targets.
Saturated. Now, what if they are heterozygous? Like they inherit a big K gene from one parent and the allele for Solano, which is little k from the other parent.
Well, now those two genes have to share that limited real estate. The cell membrane will be populated with approximately half big K antigens and half little k antigens, so the cell only has a single dose of the big K target.
And this directly impacts your serology. If you take serum containing anti-K antibodies and add it to the homozygous double-dose KK cells, you have a massive amount of targets clustered really closely together.
So the antibodies can easily cross-link the cells, right? And you get this blazing strong 3+ or 4+ agglutination reaction.
Exactly. However, if you add that exact same anti-K serum to the heterozygous KK cells, the targets are fewer and they're spaced further apart. It is physically harder for the antibodies to build a lattice.
So the visible agglutination is going to be significantly weaker, maybe only a 1+ or a 2+ reaction.
Right. That disparity in reaction strength based entirely on zygosity, that is the dosage effect.
The high-yield takeaway here for the exam is knowing exactly which blood group systems exhibit this behavior. You basically have to commit these to memory, right?
You really do. The classic systems that show dosage are M, N, S, little s. Then you have the Duffy system, PHIA and PHIB, the KID system, JK and JKB, and the antigens of the RH system, particularly big C, big E, and little e.
Okay, so connecting this to your practical bench work, when you're looking at an antibody identification panel, which is phase 3 of our roadmap, and you notice that a specific patient antibody is reacting at a 3+ strength with some cells, but only a 1+ strength with others.
Your brain should immediately start scanning for antigens that show dosage. It is an enormous diagnostic clue that helps you quickly narrow down the identity of the antibody.
Love that. Let's zoom out a bit. We've looked at the individual cell. Now we need to look at genetics across an entire population. The practice material brings up the Hardy-Weinberg formula.
Yes.
It's written out as p² plus 2pq plus q² equals 1. Now, I have to say, seeing algebra in a blood bank manual usually induces a mild panic attack for most people. Why do we need this, and what do we actually need to extract from it for the board exam?
Well, the math itself can definitely be intimidating, but the concept is actually highly practical. In population genetics, the Hardy-Weinberg principle provides a mathematical baseline to calculate the frequency of different alleles within a stable population.
But for the exam, do they actually have to calculate it?
Rarely. You rarely have to perform complex calculations, but you absolutely must understand what each variable represents.
Okay, let's ground this in a real blood group then. Let's use the M and N antigens.
Perfect. So if we assign the variable P to represent the frequency of the M gene in the population, and the variable Q to represent the N gene, the p² term in the equation mathematically represents the frequency of individuals who inherited the M gene from both parents.
So they're the homozygous MM population.
Exactly. And similarly, the Q squared term represents the homozygous NN individuals.
But the crucial piece, like the part that almost always ends up as a multiple choice question, is the middle term, right? The 2PQ.
Yes. The 2PQ represents the frequency of the heterozygous population, the individuals who inherited one M and one N, making them MN. The clinical application here is really profound.
How so?
Well, if you have a patient who has developed multiple complex antibodies and you need to find donor blood that is negative for 3 different high-frequency antigens, the blood bank uses derived Hardy-Weinberg frequencies to calculate the mathematical probability of finding that specific rare combination in the general donor pool.
So it basically tells us if we need to screen 10 units or 10,000 units.
Exactly. It sets the logistical expectation.
That makes a lot of sense. Speaking of how genes associate in a population, we really need to cover linkage disequilibrium.
This is a great one.
The source material presents this mathematical scenario. Gene X has a frequency of 0.4, and gene Y has a frequency of 0.5. Now, if inheritance was completely random, you'd just multiply them. You'd expect them to show up together in the same person 20% of the time. But observational data shows they're actually found together 35% of the time. Why does the math break down there?
The math breaks down because the physical geography of the human genome kind of overrides random statistical probability. This is what linkage disequilibrium is. It occurs when 2 distinct genes are situated extremely close to one another on the exact same chromosome.
So close that during meiosis, when chromosomes cross over and swap genetic material to create diversity, these 2 genes almost never get separated. They're a package deal.
They are genetic neighbors that simply refuse to be parted. A classic example in immunohematology is the relationship between the C and E antigens in the Rh system.
Oh, right. Because their genetic coding sequences are physically adjacent on chromosome 1.
Precisely. Because of that physical proximity, they are inherited together as a specific haplotype block far more often than individual allele frequencies would ever predict. They do not assort independently.
Okay, so let's shift from autosomes to the sex chromosomes. How does sex-linked inheritance show up on the ASCP exam? The classic anchor point here is the XJ antigen, right? The material notes that XJ is an X-linked dominant trait.
Right. And to solve an X-linked question, you just have to trace the physical path of the sex chromosomes from parent to child. It's logic. Biological sex is determined by the father. A father has one X chromosome and one Y chromosome. He must pass his Y chromosome to all of his male offspring, which is what makes them male.
And since the genetic code for the XY antigen is located exclusively on the X chromosome, none of his sons can possibly inherit the trait from him. The father-to-son transmission rate is literally 0.
Exactly, 0. Conversely, the father passes a single X chromosome to every single one of his female offspring. Because the XJ trait is genetically dominant, inheriting just that one copy from the father means every single daughter will express the XJ antigen on her red blood cells.
So on a pedigree chart on the exam, you will see an affected father with 0 affected sons but 100% affected daughters. That is the absolute hallmark of X-linked dominant inheritance. Before we leave genetics entirely, we have to touch on a population statistic that is highly testable and honestly just clinically fascinating. The Duffy frequencies. The material specifically points out the null phenotype, Phi-AB.
Yes, the Phi-AB phenotype is incredibly rare in Caucasian or Asian populations. We're talking less than 1% of individuals. However, it occurs in nearly 70% of individuals of West African descent.
70%. That is a massive statistical disparity.
It is, and it's not random. It is a textbook example of evolutionary pressure and natural selection.
Because the Duffy glycoprotein on the red blood cell isn't just some antigen we use for blood typing, right? It actually has a biological function. It acts as a chemokine receptor.
It does, and unfortunately, a specific species of malaria, Plasmodium vivax, evolved to use that exact Duffy receptor as a physical doorway to invade the red blood cell and cause infection.
So they basically hijacked the door.
Exactly. Therefore, individuals who randomly mutated to have the Phi-AB null phenotype, meaning they completely lack that specific glycoprotein doorway, were naturally immune to Plasmodium vivax malaria.
Oh, wow. So in regions like West Africa, where this specific malaria species was historically endemic, this null mutation provided a massive survival advantage.
A massive advantage. Over thousands of years, the individuals who survived or reproduced were predominantly Phi-AB, which completely shifted the genetic makeup of the entire population. It's a stunning intersection of immunohematology and infectious disease evolution. And crucially for you listening, it is a frequency disparity you must have memorized for the boards.
Absolutely. Okay, moving from the genetic blueprints to the actual weapons of the immune system, let's explore the antibodies. The practice material heavily contrasts IgM and IgG immunoglobulins.
This is foundational.
To understand their behavior in the lab, you really have to visualize their physical architecture. I always picture an IgM molecule as this microscopic, 10-armed octopus.
It's a highly accurate visualization, actually. IgM is a pentamer. It consists of 5 basic Y-shaped antibody subunits held together in a circle by a central J chain. Because it has 5 subunits, and each subunit has 2 binding sites, a single IgM molecule possesses 10 distinct antigen binding sites.
10 arms.
10 arms. It's massive. It spans approximately 35 nanometers in diameter.
And this massive size and 10-armed reach dictate exactly how it behaves in our test tubes. Because, red blood cells naturally repel each other. They really don't want to touch. But an IgM molecule is so incredibly large that it can easily span that repulsive distance between 2 separate red blood cells.
Yes, it can grab an antigen on one cell with a couple arms and just reach right across the void to grab an antigen on a completely different cell with its other arms.
And this physical bridging is what causes the visible, immediate clumping we call direct agglutination.
Precisely. This is why naturally occurring IgM antibodies, like our ABO blood group antibodies, react so vividly right there in the tube at room temperature, without any special enhancement or incubation. The pentamer structure is basically built for rapid multicellular cross-linking.
But IgG, on the other hand, is a completely different structural class entirely. It's a simple monomer. It is literally just a single Y shape. It only has 2 binding sites, and it spans a mere 14 nanometers.
It is simply too small. Under normal physiological conditions and irregular saline suspension, the repulsive gap between 2 red blood cells is physically larger than the 14 nanometer reach of an IgG molecule.
Right. So an IgG antibody can certainly recognize a foreign antigen, and it will definitely attach to that single red blood cell, which is a process we call sensitization.
Yes, sensitization.
But because it only has 2 arms and a super short reach, it just gets stuck on that one cell. It cannot bridge the gap to grab a second cell. So you end up with a tube full of red blood cells completely coated in dangerous IgG antibodies, but they aren't clumping.
To the naked eye, it looks like a perfectly smooth, negative, compatible reaction.
Which is terrifying from a clinical perspective, because if you transfuse that blood based on a false negative visual reading, those Ig-coated cells will be rapidly destroyed by the patient's spleen and liver in vivo.
And that causes a severe hemolytic transfusion reaction.
This perfectly highlights why we need chemical tools to force these tiny IgG molecules to reveal themselves. But before we get to enhancement media, the text brings up a critical behavioral difference between IgM and IgG, termed the anamnestic response.
Yes, the anamnestic response.
This is basically the mechanism behind some of the most dangerous transfusion reactions.
It is. An anamnestic response is just the immunological definition of a secondary memory response. Let's walk through the timeline. Imagine a patient is transfused with blood containing a foreign red cell antigen. Let's use the JCA antigen from the KID system for this.
Okay, the KID system. Notorious.
Right. The patient's immune system detects this foreign JCA protein and mounts a primary immune response.
Now, a primary response is sluggish, right? It takes rings to fully develop. It initially produces mostly the bulky IgM antibodies, and then it eventually transitions to producing highly specific IgG antibodies against the JCA antigen. The invading cells are cleared, the patient survives.
But the immune system learns. It generates memory B lymphocytes specifically programmed to recognize that exact JCA antigen. Now, let's say years pass. Without constant exposure to the JCA antigen, the patient's active antibody production winds down.
So the tighter the actual concentration of anti-ANTA in their plasma drops so low that it falls completely below the detection threshold of our routine blood bank laboratory screens.
Exactly. So they come into the hospital years later, we draw their blood, we run an antibody screen, and it comes up completely negative. They look like a perfectly safe, uncomplicated patient.
But they're not.
No. We cross-match what we think is compatible blood, but it happens to be positive for the JCA antigen. We hang the bag, we trigger the ambush.
The moment those JCA-positive donor cells enter the patient's circulation, those dormant memory B lymphocytes wake up, and they do not need weeks to figure out what to do this time.
They undergo rapid clonal expansion and immediately begin churning out massive quantities of highly destructive, highly specific IgG anti-JCA antibodies.
They just bypass the slow IgM phase entirely. This secondary response requires a much lower dose of antigen to trigger, and it produces an exponentially higher titer of IgG in just a matter of days.
The donor red cells that were surviving perfectly fine for the first 2 days are suddenly coated in a tidal wave of newly synthesized IgG. The patient experiences a delayed hemolytic transfusion reaction, destroying the entire transfused volume days after they were supposedly stabilized.
Understanding the anamnestic response is exactly why historical blood bank records are just as critical as current test results. You cannot ignore history. Okay, let's return to our benchwork problem. We have dangerous, small IgG antibodies that sensitize red cells but won't cause visible agglutination. How do we manipulate the physics of the test tube to force them to clump? The material details enhancement media, heavily focusing on LISS, which is low ionic strength solution, and a biophysical concept called the zeta potential.
We really must explore the physics of the red cell membrane here. Red blood cells do not just bump into each other in our veins because they are surrounded by this invisible force field of negative charge.
And this charge is primarily generated by sialic acid molecules, right? The ones embedded in the outer lipid bilayer of the cell.
Yes. In a standard saline testing environment, normal sodium chloride is highly ionized. The positively charged sodium ions in the saline are strongly attracted to that negative sialic acid on the red cell, so they rush in and form this dense physical cloud of positifications completely enveloping the red cell.
And this ionic cloud creates an electrostatic repulsion between adjacent red cells. This repulsive force is what we define as the zeta potential.
It acts exactly like two identical poles of a magnet pushing apart. And that repulsive gap is what keeps our tiny 14 nanometer IgG molecules from bridging between cells.
Enter LISS, low ionic strength solution. I mean, the name basically tells you exactly what it does.
It really does. LISS is a region where we have significantly reduced the concentration of sodium chloride. By drastically lowering the number of available charged ions in the testing environment, we essentially starve that protective ionic cloud.
The cloud shrinks.
The cloud shrinks. We have artificially lowered the zeta potential.
Because the repulsive force is diminished, the red blood cells can physically drift much closer to one another in the tube. And this proximity allows the antigens on the cell surface and the IgG antibodies in the serum to encounter each other much more frequently, vastly increasing the rate of antibody uptake and sensitization.
Yet even with LISS bringing the cells closer, sometimes the IgG molecules still can't quite form a stable lattice for visible agglutination.
And that is when we deploy the ultimate molecular bridge. Polyspecific AHG or anti-human globulin. The text specifies you really need to know exactly what is inside this reagent.
Polyspecific AHG is a marvel of applied immunology. It is literally an antibody directed against other antibodies. We inject human IgG and human complement proteins into an animal, typically a rabbit.
And the rabbit's immune system recognizes these human proteins as foreign and creates antibodies against them.
Exactly. We harvest those rabbit antibodies to make our reagent.
So polyspecific AHG contains both anti-IgG and anti-C3. C3 is a breakdown product of the complement cascade. Why is it so vital to have both in the reagent?
Detecting both is essential for capturing all mechanisms of in vivo sensitization. Sometimes an IgG antibody attaches to a red cell but doesn't activate the complement cascade. The anti-IgG component of the AHG will just grab the FCC portion of that attached human IgG, block the gap to the next cell, and cause agglutination.
Okay, but sometimes an antibody, particularly certain cold-reacting IgM antibodies, will attach to a cell, trigger the complement cascade, leave a piece of C3 permanently glued to the red cell membrane, and then physically detach and just float away.
Right, and if your AHG region only contained anti-IgG, it would completely miss this deadly immune activity.
It's a huge clinical trap.
Exactly. By including anti-C3, the AHG region can bind to those complement fragments left behind like a footprint on the cell membrane, pulling the cells together and revealing that an immune attack definitely occurred, even if the instigating antibody is no longer present. The AHG acts as the final visible link in the chain.
All right. We have established the genetic blueprints and the immunological physics. Let's move to applying them to the systems that literally dictate patient survival, the big two, the ABO and RH blood group systems. Let's start with ABO.
Okay.
I mean, it seems simple, right? AB, AB, and O.
Yep.
But the board exam goes really deep into the structural subgroups, particularly A1 versus A2. What is the fundamental difference there, both structurally and clinically?
The distinction lies in both the quantity and the physical architecture of the antigen structures on the red cell. A group A1 individual possesses an incredibly robust enzymatic pathway. They produce highly branched, highly complex A1 antigen chains, as well as simpler linear A antigen chains.
So their red cells are densely packed, expressing somewhere around 1 million antigen sites per cell.
A group A2 individual, however, has a much less efficient enzyme. They only produce the simpler linear A antigen structures, and they express far fewer of them, perhaps only 250,000 sites per cell. Crucially, they completely lack the complex branched A1 structural component.
And this structural absence drives the clinical significance. Because an A1 individual has both the A1 and the linear A structures, their immune system recognizes both forms as self. An A1 person is immunologically tolerant. They will never produce an antibody against A1.
But an A2 person's immune system has literally never seen that complex branched A1 structure. It doesn't exist anywhere in their body.
Right.
So if they were exposed to it, their immune system treats it as a foreign invader. An A2 individual has the capacity to form a naturally occurring anti-A1 antibody.
Which manifests as a classic discrepancy on the bench. When you forward type their blood, their cells react strongly with the standard anti-A region because they do possess the linear A antigens. They appear to be a normal group A.
However, the reverse typing reveals the secret. Their serum, which you expect to only agglutinate B cells, will unexpectedly and strongly agglutinate your A1 region donor cells.
You have a patient typing as A, but their serum is attacking A1 blood. You must deduce that you are dealing with an A2 subgroup who has developed an anti-A1.
To confirm this, the exam will expect you to know the specific reagent used to differentiate them. We use a plant extract, a lectin, derived from the seeds of Dolichos biflorus.
Oh, Dolichos biflorus, that name always sticks with you.
It does. This lectin has a specific structural affinity only for the complex A1 antigen. If you test the patient's cells with Dolichos biflorus and they do not agglutinate, you have confirmed they lack the A1 structure and are indeed an A2 subgroup.
Awesome. Now we arrive at a concept that absolutely blew my mind when I first encountered it. Massive high-yield flag, the Bombay phenotype.
Oh, this is a classic.
To understand how someone can have a blood type that breaks all the rules, we have to look at the biochemical assembly line, right?
Yes, the ABO antigens are not built from scratch. They are built upon a foundational precursor. You can think of it like constructing a house. You cannot put up the walls, the A or B antigens, until you have poured a solid concrete foundation. In immunohematology, that foundation is the H antigen.
And the creation of the H antigen is controlled by an entirely separate gene on chromosome 19, the FUT1 gene. If you inherit a functional FUT1 gene, it produces an enzyme that attaches a specific sugar, L-fucose, to the red cell membrane.
And that L-fucose structure is the H antigen. Once that foundation is poured, the A and B enzymes can come along and attach their specific sugars right on top of it.
The Bombay phenotype occurs when an individual inherits two recessive, non-functional H genes. Their genotype is little h. They produce zero functional enzyme. They cannot attach L-fucose. They produce absolutely no H antigen foundation.
Here is the mind-bending part. They might have inherited perfectly normal A or B genes from their parents. Those genes are churning out A or B enzymes, but those enzymes are just floating around uselessly.
Because without the H antigen foundation to attach to, the A and B sugars literally cannot be added to the cell membrane.
Therefore, when you test a Bombay individual's blood with routine anti-A and anti-B reagents, the cells do not agglutinate. The antigens just aren't there. Serologically, they phenotype exactly like a standard Group O patient.
But a normal Group O patient has massive amounts of the H antigen foundation. They just lack the A and B enzymes to build upon it. The Bombay patient lacks everything.
And that biological absence triggers a violent immunological response. Because the Bombay individual lacks the H antigen, their immune system views the H structure as a deadly foreign pathogen. They produce a naturally occurring, incredibly potent IgM anti-H antibody in addition to anti-A and anti-B.
Meaning, if you misidentify a Bombay patient as a normal Group O and you transfuse them with standard Group O packed red cells, which are absolutely covered in H antigen, their potent anti-H will trigger an immediate, catastrophic, and likely fatal intravascular hemolytic transfusion reaction.
Their serum attacks almost every unit of human blood on Earth. A Bombay individual can only ever receive a transfusion from another genetically verified Bombay individual.
That is just wild to think about. Let's address another concept that sounds almost like science fiction. The material covers the acquired B antigen. Now, wait. We just spent 20 minutes establishing that your blood type is genetically hard-coded. Your DNA dictates your transferases. How can a textbook claim a patient can suddenly acquire a new blood group antigen later in life? Doesn't that directly contradict the central dogma we just discussed?
Your skepticism is entirely justified. The patient's genetic code does not change. Their DNA remains constant. The acquired B phenomenon is an acquired serological alteration, a temporary physical modification of the red cell membrane driven by severe systemic disease. And it is exclusively observed in individuals who are genetically group A.
Okay, walk me through the pathology. How does disease alter the membrane?
It is typically associated with severe gastrointestinal pathologies, advanced colon cancer, massive bowel obstructions, or overwhelming intestinal sepsis. In these compromised states, the normal barrier of the gut breaks down, allowing massive amounts of enteric bacteria to enter the patient's bloodstream.
And these specific bacteria produce a powerful enzyme called a deacetylase.
Exactly the mechanism. The specific terminal sugar that defines the A antigen is N-acetyl-D-galactosamine. As these bacterial deacetylase enzymes circulate in the blood, they physically attack the A antigens on the red cells. They chemically cleave off the acetyl group from that complex sugar molecule.
And when you strip the acetyl group off N-acetyl-D-galactosamine, you are left with D-galactosamine. And structurally, D-galactosamine looks remarkably similar to D-galactose, which is the defining sugar of the B antigen.
The structural mimicry is so incredibly close that when you perform a routine forward typing in the blood bank, your commercial anti-B reagent gets confused. It weakly cross-reacts with this bacterially altered A antigen. The technologist sees strong agglutination with anti-A, but also unexpected weaker agglutination with anti-B.
So the patient suddenly types like an AB.
But the reverse typing saves us from the discrepancy.
Yes. The reverse typing tells the genetic truth. The patient's serum will still contain strong, naturally occurring anti-B antibodies, which will violently agglutinate regent B cells. A true AB patient would not have anti-B in their serum.
The technologist must recognize this specific clash of forward AB with a reverse A as the classic signature of an acquired B phenomena. It is a brilliant example of how systemic disease can literally hijack serological testing.
We've mentioned the sugars a few times. Let's lock them down for the exam. What are the specific immunodominant sugars that confer ABO specificity?
You must commit these molecular building blocks to memory. The foundational H antigen is defined by the addition of L-fucose. The A antigen specificity is conferred by the addition of N-acetyl-D-galactosamine. And the B antigen specificity is created by the addition of D-galactose.
In a quick, testable logic puzzle from the material, why do A1B red cells have the absolute lowest amount of unconverted H antigen on their surface?
It is a biochemical competition. On the surface of an A1B cell, you have both highly efficient A1 transferase enzymes and highly efficient B transferase enzymes operating simultaneously. Both are furiously competing to grab the available H antigen foundations and attach their respective sugars.
They're just so aggressive and efficient that they convert nearly every single H precursor chain into an A or B antigen.
Consequently, almost no unconverted raw H substance is left exposed on the membrane.
Let's shift our focus to the other half of the big two, the RH system. We've mastered ABO. How does testing for Rh antigens fundamentally differ from ABO?
The critical difference lies in the nature of the antibodies involved. As we discussed, ABO antibodies are naturally occurring, massive IgM pentamers that cause immediate visible agglutination at room temperature. They are really the exception in immunology.
The Rh system, however, operates on the standard immunological model. Rh antibodies are exposure-driven. You are not born with anti-D.
No, you only produce it if your immune system is physically exposed to the D antigen via a mismatched transfusion or through a pregnancy where fetal cells enter maternal circulation.
Because they are exposure-driven immune responses, Rh antibodies are predominantly small, monomeric IgG molecules. As we detailed earlier, IgG cannot bridge the zeta potential gap on its own.
Therefore, routine room temperature testing is often insufficient. To reliably detect Rh antigens, or antibodies directed against them, the blood bank must utilize 37 degrees Celsius incubation to optimize antibody-antigen binding kinetics, and we must deploy AHG reagent to physically cross-link the sensitized cells and force visible agglutination.
This physiological requirement brings us directly to a major benchwork procedure, weak D testing. The exam emphasizes that if a patient initially types as Rh negative at immediate spin, we cannot simply accept that result at face value.
Particularly for blood donors and prenatal patients, a simple room temperature negative is inadequate. Some individuals possess a genetic mutation that results in a significantly weakened, extremely low-density expression of the D antigen.
The antigen is physically present on the membrane, but there are just so few targets that our standard anti-D reagents cannot cross-link enough cells to create visible clumping at room temperature. They appear serologically Rh negative.
To uncover this hidden antigen, we perform the weak D test. We incubate the patient's cells with anti-D reagent at 37 degrees to allow maximal sensitization, wash the cells thoroughly, and then add AHG to build the lattice.
But the practice material sets a massive trap for students here regarding the direct antiglobulin test, the DT.
This is a critical logical constraint. The D tests whether a patient's red blood cells have already been coated with IgG antibodies in vivo, inside their own body, due to an autoimmune disease or a prior transfusion reaction.
So imagine a patient with a positive DEET. Their cells are already covered in auto-IgG. If that patient happens to have a weak D expression and you attempt a weak D test, walk me through the catastrophic bench failure.
Okay, you take their auto-coated cells, you add your commercial anti-D reagent, and you incubate. Then you wash the cells and add your AHG. The AHG molecules are designed to bind to any human IgG. In this tube, the AHG will immediately bind to the autoantibodies that were already permanently stuck to the patient's cells from the very start.
The tube will strongly agglutinate.
It will clump beautifully. And to an untrained technologist, it looks exactly like a positive weak D test. They will report the patient as Rh positive, but it is a massive false positive. The agglutination had absolutely nothing to do with the D antigen or the anti-D reagent you added. The AHG simply reacted with the patient's pre-existing autoimmune state.
Therefore, the unbreakable rule of the blood bank. If the patient's date is positive, a weak D test is completely invalid. You cannot perform it and you cannot interpret it. You must report them as Rh negative to ensure they receive safe Rh negative blood.
We must also explore another structural anomaly here, the partial D, historically known as the D mosaic. This concept answers a biological paradox that frequently appears on boards. How is it physiologically possible for an individual who types as completely Rh positive to suddenly develop a now-out anti-D antibody? Does that not violate the fundamental law of immunological self-tolerance?
It seems impossible. Like you can't make an antibody against something you already possess.
The key is that they do not possess the entire structure. The normal D antigen is not a single simple peg sticking out of the membrane. It is a massive, highly complex transmembrane protein composed of multiple distinct structural loops or epitopes. Think of it like a complex mosaic tile pattern made of 20 different colored pieces.
Okay, so most Rh-positive individuals inherited the complete blueprint and display the entire 20-piece mosaic on their red cells.
However, individuals with the partial D phenotype inherited a slightly altered genetic blueprint. They produce a D antigen, but it is physically missing one or two specific pieces of that mosaic. When we test their blood with our commercial anti-D reagents, which are really designed to recognize the overall structure, they react strongly. The bulk of the antigen is there. They type unequivocally as Rh positive.
But their immune system knows the precise microscopic topography of their own cells. Their immune system is tolerant to the 18 pieces they possess, but it has never seen the missing 2 pieces.
Exactly. So if this partial D patient receives a transfusion of normal, complete Rh positive blood, their immune system surveys those donor cells. It recognizes the 18 shared pieces as self, but it identifies those 2 extra missing pieces on the donor cells as highly foreign, dangerous pathogens.
The patient mounts a robust immune response and generates an allow anti-D specifically targeted against those missing epitopes, despite classifying serologically as Rh positive.
It is a stunning display of the immune system's microscopic precision.
Now we must confront the most dreaded section of the immunohematology curriculum, genotypes and nomenclature. We have 2 competing systems, Wiener and Fischer race. Why do we have 2, and how do we decode them without losing our minds?
Well, we have 2 systems because historically, the scientists who discovered the Rh system fundamentally disagreed on how it was actually inherited. Fischer and Race believed the Rh system was controlled by 3 closely linked separate genes, D, C, and E. Their nomenclature is straightforward and uses those specific letters.
Alexander Wiener, however, believed the entire complex was controlled by a single master gene locus that produced a massive multi-part agglutinogen structure. He created this incredibly complex shorthand using Rs, primes, and numbers to describe the entire inherited package, the haplotype.
Modern molecular biology proved that Fischer and Race were closer to the truth. It's actually 2 linked genes, one for D and one that codes for both C and E. However, Wiener's shorthand was so deeply ingrained in medical literature that we still use it today. You absolutely must be able to translate between them. The strategy is to establish a mental decoding key.
Let's build the key based on the practice material. First rule, the presence or absence of the D antigen.
In Wiener shorthand, any capital R definitively indicates the presence of the D antigen. The haplotype is R chest positive. Conversely, any lowercase r indicates the complete absence of the D antigen. The haplotype is Rh negative.
Second rule, decoding the C and E antigens.
This involves the superscripts. Let's look at the Rh positive R haplotypes. R0 translates to Ds. It is the baseline, no superscripts. R1 translates to DC. A helpful mnemonic is to visualize the number 1 as a tall, straight vertical line. It looks somewhat like a capital C. So R1 has the big C. R2 translates to DCE.
Now let's apply the rules to the Rh negative lowercase r haplotypes.
The baseline lowercase r translates to Ds. This is the classic complete absence of major Rh antigens. An R with a single prime marquee, prime indicates the presence of big C, so R is DIC. An R with a double prime mark, R double prime, indicates the presence of big E, so RA is DCE.
The text also throws in some older specific antigen translations that still haunt exams. RHR prime refers specifically to the little c antigen. RH double prime refers to the big E antigen. Let's apply this translation key to a complex clinical scenario provided in the text. You have a patient whose immune system is highly sensitized. They have formed 3 distinct allo antibodies, anti-C, anti-E, and anti-D. As the technologist, what precise genotype of blood must you secure to ensure a safe transfusion?
You must find donor units that completely lack all 3 of those specific target antigens. The donor cells must not have big C, they must not have big E, and they must not have D. You need blood that is purely little d, little c, little e.
And converting that vital requirement into Wiener shorthand to search the donor database.
You are looking for the RA genotype. This is the baseline Rh negative haplotype inherited from both parents. Fortunately for this patient, R is the most common Rh negative genotype globally, occurring in roughly 14% of Caucasian donors. It is perfectly safe because it entirely lacks the D, big C, and big E targets, meaning the patient's circulating antibodies will have absolutely nothing to attack.
Awesome. We have completed the genetic and systemic foundations. We are now moving into segment 3, the detectives. We have run an antibody screen on a pre-op patient, and it is positive. We have detected a rogue IgG molecule. Now, we must perform an antibody identification panel to literally name the threat. This requires strict statistical and procedural rules. Let's examine the foundational law of antibody ID, the 3x3 rule.
In immunohematology, we cannot rely on a single positive reaction to confirm an antibody's identity. Biological variability is simply too high. We require a statistical certainty of 95%, which corresponds to a p-value of 0.05. To achieve this mathematical confidence, the 3x3 rule mandates 2 distinct criteria.
First, the inclusion criteria.
You must identify at least 3 separate donor red cell panel members that possess the suspected antigen. When tested, the patient's serum must react positively with all 3 of those cells.
Second, the exclusion criteria.
You must identify at least 3 separate panel members that entirely lack the suspected antigen. The patient's serum must not react with any of these 3 cells. This binary confirmation, proving that the antibody reliably fires when the specific target is present and dependably holds its fire when the target is absent, is the only way to clinically prove identity.
But the practice material highlights a massive, dangerous caveat when you are crossing out or ruling out antibodies that you believe are not present in the patient's serum.
The rule-out process is incredibly fraught with peril due to the dosage effect we discussed earlier. To confidently strike an antibody off your suspect list, you must use a panel cell that is homozygous for the target antigen. The cell must possess a double dose. And of course, the patient's serum must yield a completely negative reaction against that saturated cell.
What happens if you try to take a shortcut and use a heterozygous single-dose cell to rule out an antibody?
You risk a catastrophic false negative. If the patient has a weak, low-titer antibody in their plasma, it might not be potent enough to create visible agglutination when confronted with the sparse single-dose antigen distribution on a heterozygous cell. The tube looks negative.
You document a negative reaction, you cross that antibody off your list, and you clear the patient for surgery.
Exactly the nightmare scenario. You have conceptually erased an antibody that is actually actively lurking in the patient's plasma. If the blood bank then issues a donor unit that happens to be homozygous for that antigen, the sudden influx of dense targets will trigger the weak antibody into a massive, destructive hemolytic reaction. You must use double-dose cells to guarantee the absence of a threat.
Let's discuss the clinical reality of high versus low-frequency antigens. The text specifically highlights the K antigen, also known as Telano, from the KEL system.
Telano is categorized as a high-frequency antigen. It is physically present on the red blood cells of 99.8% of the global human population. This statistical dominance creates a fascinating inverse clinical dynamic.
Because almost every human being on the planet possesses the Solano antigen, their immune systems recognize it as self. Therefore, almost no one ever forms an anti-anti antibody. Finding a patient with anti-anti is an incredibly rare event.
However, if you do encounter that 0.2% patient, someone who genetically lacks Solano, was exposed to it via prior transfusion, and actively formed an anti-antidecal antibody, you are facing a blood bank crisis. Finding compatible blood for this patient is mathematically terrifying. 998 out of every 1,000 randomly selected donor units will be incompatible and will cause a reaction.
You can't just pull units off the shelf and cross-match them. You'd waste days. How do you actually solve this for a bleeding patient?
You immediately bypass local inventory. You have to access national or international rare donor registries, locating frozen units from the microscopic fraction of donors who share this rare genetic absence. If the surgery is elective, the best option is often autologous donation, where the patient donates and stores their own blood weeks in advance. Alternatively, you test the patient's immediate siblings, as they have a 25% statistical chance of inheriting the same rare genetic combination.
Let's bring out the heavy chemical tools. Enzyme treatments. This is high H yield for problem-solving panels. We talked about how less changes the electrical charge. Enzymes are much more violent. They physically demolish structures on the cell membrane.
We utilize proteolytic enzymes, primarily physin derived from figs or papain derived from papaya. These enzymes do not just alter the charge, they physically cleave peptide bonds. They take molecular scissors to the complex glycoproteins protruding from the red cell lipid bilayer. When you run an antibody panel using enzyme-treated cells, you are looking for dramatic shifts in reaction strength to guide your diagnosis.
You categorize them into the enhancers and the destroyers. Let's start with how enzymes act as enhancers.
Enzymes dramatically enhance the reactivity of antibodies directed against the Rh system and the KID system, so JK and JKB. The Rh antigens, for example, are non-glycosylated transmembrane proteins that sit very tightly embedded within the lipid bilayer, often partially obscured by neighboring bulky glycoproteins.
When you apply the enzyme, it acts like a lawnmower.
Precisely. It shears off those surrounding bulky glycoproteins. It removes massive amounts of steric hindrance, deeply exposing the Rh proteins. Additionally, by shearing off glycoproteins that carry sialic acid, it drastically reduces the zeta potential. The combination of exposed targets and reduced repulsion allows Rh and KIT antibodies to bind with devastating efficiency. A weak 1+ reaction on a standard panel will explode into a blazing 4+ reaction on an enzyme panel.
But those molecular scissors are indiscriminate. What do they destroy?
Enzymes completely obliterate the physical antigens of the Duffy system, Phia and Fiabin, the MNS system. The specific epitopes that define these antigens are located on the distal ends of very long, fragile glycoproteins. The enzymes physically slice those glycoproteins off at the base. The structural targets are quite literally washed down the sink during the preparation phase.
Let's put this into a study scenario. You are running a complex panel. You see multiple varied reactions at the HG phase. You suspect multiple antibodies. To clarify the picture, you run an enzyme-treated panel using the exact same donor cells. Suddenly, 3 of your previously positive reactions vanish. The tubes are completely clear. What is your deduction?
The disappearance of reactivity upon enzyme treatment is the diagnostic hallmark of an antibody directed against the Duffy or MNS systems. Because the enzyme destroyed the target, the antibody has nothing left to bind to. Conversely, if a reaction remains and becomes significantly stronger, you have confirmed the presence of a resilient, enhanced antibody strongly pointing toward Rh or KID.
Let's do a deeper dive into the specific behavioral quirks of various antibody systems detailed in the material. These are the weird exceptions that define the immunohematology exams. Let's start with the Lewis system, specifically Leah and Leb.
The Lewis system is unique because the antigens are not intrinsic structural components of the red blood cell membrane. They're actually manufactured by tissue cells in the gut and salivary glands, secreted into the plasma as glycolipids, and then they circulate and passively adsorb. They basically stick onto the surface of the red blood cells.
Because they are just sticking to the outside, Lewis antibodies are generally considered a clinical nuisance. They're usually naturally occurring, cold-reacting IgM molecules that detach at 37 degrees and rarely cause actual red cell destruction in vivo.
However, because Lewis antigens exist in high concentrations in soluble form within body fluids, we can use a chemical trick to confirm their identity. If a patient's serum contains anti-Leb, We can neutralize that antibody in the test tube by adding human saliva that is rich in soluble Lewis substance. The soluble antigens in the saliva bind up all the antibody in the serum, rendering it completely inert. When you then test that neutralized serum against lead-positive red cells, the reaction will disappear.
But the material specifies a genetic lock on this process. Not everyone secretes Lewis substance into their saliva.
Correct. The production of soluble Leb substance requires a complex genetic interplay. The patient must inherit 3 distinct dominant functional genes: the Lewis gene to make the basic antigen, the H gene to provide the precursor structure, and the secretor gene, which specifically acts as the switch, allowing these antigens to be secreted into bodily fluids. If they lack any of these 3, they cannot neutralize the antibody. The text also wisely notes that when performing this neutralization, you should use saliva from a group O individual to avoid introducing soluble A or B antigens that could cause cross-reactivity and confuse the results. Next quirk, the P system. Anti-P1 is another commonly encountered, usually insignificant, cold-reacting IgM antibody. Much like the Lewis system, it can be neutralized. However, the soluble P1 substance is not found in human saliva. To neutralize anti-P1, the blood bank must use hydated cyst fluid derived from tapeworm infections or an extract from pigeon egg whites, both of which contain structurally identical P1-like glycoproteins.
But the P system also has a terrifying, highly pathogenic side. The practice material specifically links the P system to paroxysmal cold hemoglobinuria, or PCH.
PCH is a severe autoimmune hemolytic anemia caused by the Doneth-Lansdyner antibody. This is not a harmless cold IgM. It is a highly destructive biphasic IgG autoantibody directed against the P antigen on the patient's own red cells.
Biphasic meaning it requires 2 distinct temperature phases to execute its destruction. Walk us through the pathology.
It operates like a delayed fuse bomb. When the patient is exposed to cold temperatures, perhaps walking outside in winter, the blood circulating in the colder extremities, like the fingers, toes, or earlobes, drops below 37 degrees. In this cooler environment, the Doneth-Lansdyner IgG antibody binds tightly to the P antigens on the red cells. It sensitizes them. Furthermore, it initiates the early stages of the complement cascade.
But it doesn't cause immediate hemolysis in the fingertips.
No, the complement cascade requires thermal energy to complete its final destructive sequence. As that cold, sensitized blood circulates back into the warm core of the body, the heart, the lungs, the deep organs, the temperature rises back to 37 degrees. This sudden warming violently accelerates the complement cascade. The membrane attack complex forms, punching massive physical holes in the red blood cells, leading to catastrophic, sudden intravascular hemolysis and the release of free hemoglobin into the urine, hence the name cold hemoglobinuria.
It is a stunning mechanical process. Next on the list, the I system, big I versus little I. The text frames this as a developmental antigen.
It is an antigen system that physically changes architecture as a human ages. The little I antigen is a straight, linear carbohydrate chain. Red blood cells from fetuses and newborns' cord blood are heavily populated almost exclusively with this linear little I antigen. They possess very little branch structure.
But as the infant grows into an adult, a branching enzyme activates.
Yes. Over the first 2 years of life, an enzyme begins adding complex carbohydrate branches to those linear chains. This branch structure is defined as the big I antigen. Therefore, normal adult red blood cells are rich in big I and have almost completely lost their little I expression.
This biological shift is the key to identifying a common nuisance antibody.
Anti-I is a frequent cold autoantibody. It will react with almost every single cell on a standard adult antibody panel because all adults have the big I antigen. The diagnostic trick is to run the patient's serum against a sample of O-negative cord blood. Because the cord blood lacks the big I architecture, the antibody cannot bind. A strong positive reaction against all adult cells coupled with a completely negative reaction against cord blood is the definitive proof of anti-I.
What about the KID system, JCO and JKB? We mentioned them earlier when discussing the anamnestic response. The text paints them as the villains of the blood bank.
They are notorious assassins because they behave like immunological sleeper agents. KID antibodies are infamous for their ephemeral nature. After a primary exposure, their titers drop rapidly in vivo, often falling well below the detection limits of our most sensitive laboratory screens. The patient's serum tests cleanly negative.
You clear them for transfusion, you hang the JAK positive unit, and the memory B cells wake up.
The rapid, massive secondary production of IgG anti-JICA triggers a severe delayed hemolytic transfusion reaction, destroying the unit days after the event. Furthermore, Kit antibodies are notorious for demonstrating severe dosage effects, making them incredibly difficult to identify during their weak phases. As we noted, they're strongly enhanced by enzymes, making an enzyme panel a critical tool when you suspect a fading kit antibody.
The final quirk the text connects is warm autoimmune hemolytic anemia, WAIAA, to a very specific antibody target.
WAIAA occurs when a patient's immune system loses tolerance and creates an IgG autoantibody that reacts at body temperature, constantly destroying their own red cells. While these autoantibodies can be incredibly complex, they're frequently directed against the core Rh protein complex, specifically targeting the little e antigen. Because little e is a high-frequency antigen present in 98% of the population, this autoantibody will indiscriminately agglutinate nearly every single donor unit you cross-match, creating a massive logistical hurdle for transfusion support.
Let's finish the detective segment with a discrepancy troubleshooting scenario from the practice material. You are typing a new patient. You perform the forward typing mixing the patient's wash cells with your commercial anti-A and anti-B reagents. The tubes instantly clump. It is a solid 4+ positive for everything. They look like an AB.
Okay, an AB phenotype. Proceed to the reverse.
You perform the reverse typing, mixing the patient's unwashed serum with your commercial A1 and B cells. Those tubes also instantly clump. It is a solid positive for everything. They look like an O. You have a massive fundamental clash. The forward says AB, the reverse says O. What biophysical mechanism is breaking the test, and how do we resolve it on the bench?
When every single tube in an ABO typing is universally positive at room temperature, you must immediately suspect the presence of a highly potent, high-titer cold autoantibody. This autoantibody, often an anti-I as we just discussed, is non-specifically binding to every single red cell in the testing environment, both the patient's own cells and the reagent cells, causing massive, indiscriminate clumping that completely masks the true ABO reactions.
So the reactions we're seeing have nothing to do with A or B antigens. It is just autoimmune static. How do we clear the static?
We must physically separate the autoantibody from the testing components. Let's fix the forward typing first. The forward typing requires the patient's red blood cells. Currently, those cells are thickly coated with the cold IgM autoantibody. To remove it, you must wash the patient's cells multiple times using saline that has been preheated to 37 degrees Celsius.
The thermal energy breaks the weak hydrogen bonds holding the cold IgM to the cell membrane. The antibody elutes it physically falls off into the saline wash, which you discard.
Exactly. You are left with a clean suspension of the patient's red cells, free of autoimmune interference. You can now test those warm wash cells with anti-A and anti-B to reveal their true forward blood type.
Now we have to fix the reverse typing, which requires the patient's serum. The serum is currently saturated with the cold autoantibody. Heating the serum won't help. The antibody is still floating in there and will bind to our reagent cells the moment it hits room temperature.
To clear the serum, we must perform a procedure called an autoadsorption. This is essentially using the patient's own red blood cells as a biological sponge. We take a fresh aliquot of the patient's serum and mix it with a large volume of their own thoroughly washed red cells. We incubate this mixture at 4 degrees Celsius, the optimal temperature for the cold autoantibody to bind.
The autoantibody in the serum violently attacks and binds to the massive influx of the patient's own red cells.
Once the binding is complete, we place the tube in a refrigerated centrifuge and spin it down. The heavy red cells, now completely saturated with the extracted autoantibody, form a dense pellet at the bottom of the tube. We carefully pipette off the clear serum sitting on top. This newly harvested serum has been physically stripped of the interfering autoantibody. We can now use this adsorbed serum against our A1 and B cells to obtain a valid, accurate reverse typing.
We are moving to segment 4, the matchmakers. Armed with all our genetic rules and detective work, we arrive at compatibility testing. How does the laboratory actually select, test, and legally clear a unit of blood to be hung on a patient's IV pole? Let's start with the cornerstone, the major cross-match.
The major cross-match is the absolute final serological safety check before blood enters a human body. It is an in vitro simulation designed to mimic exactly what will happen in vivo upon transfusion. The strict, unyielding definition of a major cross-match is that it always uses the recipient's serum tested against a physical sample of the donor's red blood cells.
We are asking one specific physiological question. Does this patient have circulating antibodies in their plasma that will immediately attack, bind to, and destroy this specific incoming unit of donor red cells?
That is the only question that matters for immediate survival. Now, a key conceptual point often tested on the board exam. What happens if the situation is reversed? What if the donor unit happens to contain an unexpected antibody? Let's say the donor has a strong anti-Kel circulating in their plasma. Does that donor antibody ruin the major crossmatch?
Logically, no, because we don't even use the donor's plasma in the major cross-match procedure. We only wash and use their red cells.
Precisely. But even in vivo, during the actual transfusion, it's rarely a significant clinical problem. The primary product we transfuse today is packed red blood cells. The vast majority of the donor's plasma has been removed during processing. The tiny volume of residual donor plasma and whatever antibodies it contains is infused into the patient and immediately diluted into the patient's entire 5-liter circulating blood volume. The antibody titer is dispersed so rapidly that it cannot mount a coordinated attack on the patient's cells. The overwhelming primary threat is always the recipient's massive plasma volume attacking the concentrated incoming donor cells.
Now, in modern high-volume laboratories, the physical test tube major crossmatch has largely been replaced by the electronic or computer crossmatch. But the material stresses that you cannot just click a button. The FDA and blood bank standards mandate incredibly strict high-yield constraints before a computer can clear a unit.
The electronic crossmatch relies entirely on the absolute accuracy of the data entered and the rigid logic programmed into the system. First, the software must be coded with hard stops that physically prevent a technologist from assigning or releasing an ABO-incompatible unit. The computer must throw an override-proof error if you try to give A blood to an O patient.
But the computer is only as smart as the data you feed it. The second constraint is the vital safety net.
To perform a computer crossmatch, there must be 2 separate concordant determinations of the recipient's ABO blood type securely recorded in the laboratory information system. One of those determinations must be from the current, freshly drawn sample for this specific hospital admission. The second determination can be a verified historical record from a previous admission, or it must be a completely separate second blood draw taken by a different phlebotomist at a different time.
Why require 2 separate draws? What physiological problem does that solve?
It doesn't solve a physiological problem, it solves a human one. It exists solely to prevent the single most common cause of fatal hemolytic transfusion reactions globally: WBIT, or wrong blood in tube. A nurse draws blood from Mr. Smith, but accidentally slaps Mr. Jones' label on the vial. If the computer relies on just that one mislabeled tube, it will cross-match blood for the wrong patient, leading to a catastrophic reaction. By requiring 2 separate testing events to perfectly match, you mathematically drastically reduce the odds of a fatal clerical error slipping through.
If the historical record says the patient is A positive, but the new mislabeled tube types as O positive, the computer logic immediately locks down and refuses to cross-match anything until a manual, rigorous investigation resolves the discrepancy. Let's move to emergency scenarios. Universal donors and emergency release. A trauma patient is bleeding out in the ER. We don't have 45 minutes to run a full antibody screen and cross-match.
In massive exsanguination, oxygen delivery supersedes serological perfection.
Yeah.
We immediately issue O-negative packed red blood cells. We call O-negative the universal donor because the red cells physically lack the A antigen, the B antigen, and the highly immunogenic D antigen. Regardless of the patient's blood type, they should not have naturally occurring antibodies against O-negative cells.
But the practice material presents a specific nightmare massive transfusion scenario. You are at a small regional hospital. A multi-vehicle trauma comes in. You exhaust your entire inventory of O-negative blood in the first hour. The bleeding patient is a male. A rapid typing reveals his blood type is AB negative. His antibody screen is negative. You must issue more blood immediately, but you only have Group A, Group B, and Group O positive units left. The text explicitly states that A positive or A negative units are preferred over B or O. Why on earth would you intentionally give an AB patient Group A blood instead of Group O?
It seems incredibly counterintuitive, but it relies on a deep understanding of plasma immunogenicity. Let's break down the patient. He is Group AB. This means his own red blood cells possess both the A and the B antigen structures. Consequently, his immune system is tolerant to both. He does not produce anti-A or anti-B. Therefore, transfusing him with A, B, or O red cells is technically perfectly safe from a cellular compatibility standpoint. His body will accept all of them.
The danger isn't the donor cells. The danger is what comes with the donor cells.
Exactly. Even packed red blood cell units contain roughly 20 to 30 milliliters of residual donor plasma. If you issue Group O units, you are injecting residual plasma that contains high titers of both anti-A and anti-B antibodies. If you issue Group B units, you are injecting plasma rich in anti-A. If you give 1 or 2 units, dilution handles it. But in a massive transfusion protocol where you might give 20 units in an hour, that residual donor plasma accumulates rapidly in the patient's circulation.
The donor plasma builds up to a critical titer and begins actively attacking the patient's own AB red blood cells.
It causes a massive iatrogenic hemolytic reaction. The text specifies Group A as the preference because immunological studies indicate that naturally occurring anti-B found in Group A plasma is generally less potent and less destructive than anti-A found in Group B or O plasma. By giving Group A units, you are only introducing the weaker anti-B into the patient's system, minimizing the collateral damage to his own red cells.
And what about the Rh factor? You are giving Rh positive blood to an Rh negative patient. You are intentionally immunizing him.
In emergency trauma, you balance risk. The patient is male. If he forms an anti-D antibody later, he cannot suffer from hemolytic disease of the fetus and newborn. It simply means he will require Rh negative blood for future elective surgeries. Saving his life today from hemorrhagic shock is worth the manageable complication of future immunization.
That is some high-level strategic blood bank chess. Let's return to standard non-emergency cross-matching. We have two physical methods, immediate spin versus AHG cross-match. What dictates which method the technologist is legally allowed to perform?
It depends entirely on the patient's immunological history. The immediate spin, or IS cross-match, is rapid. You mix patient serum and donor cells, spin it in the centrifuge for 15 seconds, and read it. However, it only verifies gross ABO compatibility at room temperature. You are only permitted to perform an IS cross-match if the patient meets two strict criteria. Their current antibody screen is unequivocally negative, and a thorough review of their historical record shows no evidence of ever having a clinically significant, unexpected antibody.
If they fail either criteria, if their screen is positive today, or if a record from 10 years ago shows they once had an anti-J-Strixaw that is currently undetectable, you cannot use immediate spin.
You must perform a full AHG cross-match. You must incubate the patient's serum and the donor cells at 37 degrees for an extended period. Wash them, and apply AHG reagent. You must physically prove that the patient's historical or current IgG antibodies will not attack that specific donor unit at physiological body temperatures.
Let's do some cross-match troubleshooting based on the text. Scenario 1. You are running a full AHG cross-match. The screening cells are positive. The cross-match tube with the donor unit is positive. And crucially, the autocontrol, where you test the patient's serum against their own washed red cells, is also glaringly positive. The agglutination persists through immediate spin, 37 degrees, and the AHG phase.
The autocontrol is the diagnostic key here. If the patient's serum is actively attacking their own red cells at all thermal phases, it heavily points away from a simple allo antibody directed against a foreign donor antigen. It strongly suggests a systemic issue. You're likely dealing with a potent, nonspecific, warm autoantibody, like WIIA, that is coating every cell in the testing environment. Alternatively, the patient may have a severe hyperproteinemia, an abnormal excess of plasma proteins that is causing nonspecific chemical clumping.
Scenario 2. You perform an immediate spin cross-match. The tube is heavily clumped. It looks completely incompatible. You decide to incubate it at 37 degrees, wash it 3 times with saline, and run it through the AHG phase. After washing and adding AHG, the incompatibility completely vanishes. The tube is crystal clear and perfectly compatible. What biophysical illusion just happened?
You just observed the classic serological hallmark of rouleau. Rouleau is not true immunological agglutination. It is a physical stacking of red blood cells, resembling a stack of coins. This stacking is caused by massive excess of heavy proteins in the patient's plasma, most commonly seen in patients with multiple myeloma or Waldenstrom's macroglobulinemia.
The heavy proteins alter the dielectric constant of the plasma, overwhelming the zeta potential and physically pushing the cells together.
Precisely. At room temperature immediate spin, this heavy protein stacking looks visually identical to an IgM antibody reaction. You cannot tell the difference with the naked eye. But the key is the washing step required before the AHG phase. During those 3 vigorous saline washes, you are physically flushing away all of that excess paraprotein. With the heavy proteins gone, the zeta potential reasserts itself, the coin stacks break apart, and the cells resuspend normally. The AHG reaction is negative because there was never an actual antibody attached to the cells.
The practice material provides a specific benchwork fix for this, the saline replacement technique. If you suspect rouleau at immediate spin, you don't wait for the AHG phase. You take the clumped tube, spin it hard in the centrifuge to pack the cells at the bottom. You carefully pipette off the patient's protein-rich serum and replace it with 2 drops of normal saline. You mix and respin.
A perfectly executed serological trick. If the initial clumping was caused by a true bound IgM antibody, Replacing the serum with saline won't break the strong lattice. The clumps will remain. But if it was merely rouleau protein stacking, the saline physically disperses the proteins, the stacking collapses, and the false positive reaction completely disappears, proving the unit is actually compatible.
One final absolute mandate from this section: sample retention.
Blood bank standards dictate that the recipient's pre-transfusion blood sample, the exact tube used for the cross-match, must be tightly sealed and securely stored in a monitored refrigerator for an absolute minimum of 7 days post-transfusion. This is not a recommendation. It is vital forensic evidence. If the patient suffers a delayed hemolytic reaction 5 days after discharge, the blood bank must have that original pre-transfusion sample available to retest, reevaluate, and determine exactly how the antibody was missed or how the immune system shifted.
We have reached segment 5, the fallout. We have done the testing, we have issued the blood. What happens when the biological systems violently reject our matchmaking? What happens when the immune system attacks a transfusion, or more tragically, attacks a developing fetus? Let's start with transfusion reactions. The material is incredibly clear on step 1. If a nurse calls the blood bank from the ICU and says, "My patient is having a severe reaction to the blood," what is the very first immediate action the technologist must take?
The absolute first step is to stop the transfusion, maintain IV access, and immediately check for clerical errors. We touched on this during the computer crossmatch, but it is the paramount rule of transfusion medicine. Roughly 90% of all acute, severe hemolytic transfusion reactions are not caused by complex, undetectable, exotic antibodies. They are caused by simple, devastating human error.
The wrong unit of blood was hung on the wrong patient.
Yes. The technologist must immediately verify all tags, wristbands, unit numbers, and paperwork. Simultaneously, the clinical team must draw a new post-transfusion blood sample from the patient and send it to the lab. The technologist will centrifuge this new sample and visually inspect the serum. Is it a normal straw color, or is it bright pink or red?
Pink or red serum indicates massive intravascular hemolysis. The red cells are exploding inside the blood vessels, releasing free hemoglobin directly into the plasma.
Exactly. The technologist then repeats the ABO and RH typing on this new sample to ensure it matches the pre-transfusion records. Finally, and most critically, they perform a direct anti-globulin test at DEET on the post-transfusion red cells
to see if the circulating donor cells have been coated with the patient's antibodies during the reaction.
Correct. A newly positive DEET is definitive serological proof that an immune-mediated attack is actively occurring in vivo. Let's break down the specific clinical pathologies of the reactions the text covers. First, anaphylactic or anaphylactoid reactions.
These are terrifying, sudden, severe, life-threatening allergic reactions characterized by rapid respiratory distress, bronchospasm, and hypotensive shock occurring within minutes of the transfusion starting. What is the specific molecular pathophysiology here?
It is a severe hypersensitivity reaction, and it occurs almost exclusively in a very specific patient population. Individuals who possess a genetic profound deficiency in IgA immunoglobulins. Because their bodies have never produced normal IgA, their immune systems have never developed tolerance to it. Over time, through environmental exposure, they form highly potent circulating anti-IgA antibodies.
When we transfuse them with a standard unit of packed red cells, that unit contains roughly 20 milliliters of normal donor plasma, and normal donor plasma is packed with IgA.
The moment that donor IgA hits their bloodstream, their massive anti-IgA titers attack it, triggering a catastrophic systemic release of histamine and vasoactive mediators. It is pure anaphylaxis.
The treatment for the acute event is epinephrine. But what is the preventative strategy for future transfusions? We can't just cross-match for IgA.
We cannot use standard units. These patients must be transfused with cellular products that have been subjected to an intensive washing protocol. The packed red cells are washed multiple times with massive volumes of saline in an automated cell washer to completely strip away every last microscopic trace of donor plasma and residual IgA proteins, rendering the cells safe for infusion.
Next reaction, TRALI. Transfusion-related acute lung injury. This is currently one of the leading causes of transfusion-related morbidity and mortality. The patient develops severe acute respiratory distress, hypoxemia, and bilateral pulmonary edema. Their lungs rapidly fill with fluid, usually within 6 hours of the transfusion.
The mechanism of TRALI is fascinating because it is essentially the exact opposite of a standard hemolytic reaction. In a normal reaction, the recipient's massive plasma volume attacks the incoming donor cells. In TRALI, the attack comes from the bag. The donor's plasma contains incredibly potent, preformed antibodies directed specifically against human leukocyte antigens, HLA, or specific human neutrophil antigens, HNA.
So the weapon is injected into the patient.
Yes. When this antibody-rich donor plasma enters the recipient, the donor antibodies immediately seek out the recipient's white blood cells, specifically the neutrophils. This collision occurs primarily in the dense microscopic capillary beds of the patient's lungs. The donor antibodies violently activate the recipient's neutrophils.
The activated neutrophils degranulate, releasing toxic enzymes and reactive oxygen species directly into the delicate pulmonary endothelium.
The capillary walls are shredded, massive inflammation occurs, the endothelial barrier fails, and plasma fluid leaks uncontrollably into the alveolar air spaces, causing the sudden, life-threatening pulmonary edema.
Because these HLA antibodies are usually formed through exposure to foreign white cells during multiple pregnancies, blood collection centers actively mitigate Chi-LALI risk by diverting plasma donations from multiparous women away from direct clinical transfusion, routing it instead for pharmaceutical manufacturing. Let's move to a milder but far more common reaction, the febrile non-hemolytic transfusion reaction.
This is clinically defined as an unexplained rise in patient body temperature of at least 1 degree Celsius during or shortly after the transfusion, accompanied by chills or rigors, but with absolutely no serological or physical evidence of hemolysis.
The red cells are fine, something else is causing the fever.
It is typically driven by two distinct mechanisms. First, the recipient may have pre-existing antibodies directed against the residual donor white blood cells, leukocytes, that were inadvertently packed into the unit. When the antibodies attack the donor white cells, it triggers a febrile immune response. Second, during prolonged refrigerated storage, those passenger donor white cells actively metabolize and secrete inflammatory cytokines directly into the plasma of the bag. When you infuse the bag, you infuse a concentrated dose of fever-inducing chemicals.
The solution here is straightforward and preventative: leukocyte-reduced blood products. By filtering the unit immediately after collection, you physically remove 99.9% of the donor white blood cells, eliminating both the antibody targets and the source of the accumulative cytokines. Now, let's officially define the clinical manifestation of the anamnestic response we discussed earlier, the delayed hemolytic transfusion reaction.
The timeline is the defining feature. The patient is transfused with fully compatible cross-matched blood. They tolerate the procedure perfectly. But days, sometimes a week later, they present with an unexplained steady drop in hemoglobin, mild clinical jaundice, and a low-grade fever. The culprit is almost always an antibody that evaded detection, most commonly from the KID system, like anti-JKB.
The re-exposure to the JKB antigen on the donor cells triggered the memory B cells.
Yes, the newly synthesized IgG anti-JKB floods the plasma. However, unlike a violent ABO mismatch, IgG typically does not activate complement all the way to completion. Instead, the IgG coats the donor red cells. These coated cells circulate until they pass through the incredibly tight capillary networks of the spleen. The macrophages in the spleen recognize the IgG coating, grab the red cells, and physically destroy them. This is extravascular hemolysis. It is slower, steadier, and less dramatically fatal than intravascular hemolysis, but it ruins the therapeutic benefit of the transfusion.
And the serological proof. If you draw their blood 5 days later and run a date, what do you see under the microscope?
The DAT will be strongly positive for IgG, but the microscopic field will look bizarre. You will observe a classic mixed field agglutination pattern. You will see tight, dense clumps of agglutinated red cells flowing in a massive sea of completely free, unagglutinated cells.
Explain the physics of the mixed field. Why isn't everything clumping?
Because of the precise specificity of the antibody. The patient's newly formed anti-JKB is only attaching to the surviving transfused donor cells that possess the JKB antigen. It is ignoring the patient's own native red cells, which are genetically JKB negative. When you add AHG region, only the coded donor cells are pulled into clumps. The massive sea of unagglutinated cells represents the patient's own unharmed blood volume. It is visual proof of an ongoing, highly targeted immune clearance.
Let's shift to the final and perhaps most emotionally weighty topic in the practice material: hemolytic disease of the fetus and newborn, or HDFN. What is the fundamental biological mechanism driving this pathology?
HDFN occurs when a pregnant woman's immune system launches a targeted attack against the red blood cells of her developing fetus. It requires a specific set of circumstances. First, the mother must possess a circulating IgG alone antibody directed against a specific red cell antigen. Second, she must lack that antigen herself, but her fetus must have inherited the gene for that exact antigen from the biological father.
The critical physical property here is the size and structure of the antibody.
Exactly. Large IgM antibodies like natural anti-LEO or anti-P1 cannot physically cross the placental barrier, but IgG is a small monomer. Furthermore, the placenta possesses specific Fc receptors that actively grab maternal IgG from her bloodstream and actively transport it across the barrier into the fetal circulation to provide the fetus with passive immunity.
But the placenta doesn't know the difference between helpful Ig against tetanus and harmful IgG against the fetal red cells.
It transports everything. The maternal IgG enters the fetal bloodstream, identifies the target antigens on the fetal red blood cells, and binds tightly to them. These sensitized fetal cells are then rapidly destroyed by the fetal spleen. This massive clearance causes profound fetal anemia. To compensate, the fetal liver and spleen enlarge massively to accelerate red cell production, often failing and leading to heart failure and hydrox fetalis, severe fluid accumulation, which can cause death in utero. If the infant survives to term, the rapid breakdown of red cells after birth overwhelms their immature liver, leading to severe jaundice and the deposit of toxic bilirubin in the brain, causing permanent neurological damage known as kernicterus.
The moment a baby is born to a mother with a known antibody, the lab immediately runs a DAT on the umbilical cord blood to see if the cells are actually coded. The text notes the cord DAT will be strongly positive if coded with maternal immune anti-D, anti-K, or anti-C. But here is a critical logical note from the host side to watch out for on the exam. The baby must possess the target.
An essential constraint.
If the mother is genetically K-negative and she has formed a massive titer of anti-Kel IgG from a previous transfusion, her body is full of the weapon. But if the father is also K-negative, the baby will be K-negative.
In that scenario, the mother's anti-Kel Ig will still be actively transported across the placenta. Her antibodies will completely flood the fetal circulation, but because the fetal red cells entirely lack the Kel antigen, the antibodies have absolutely nothing to bind to. They will circulate harmlessly, the infant is perfectly safe, the cord blood PD will be stone cold negative, and no clinical intervention is required. The weapon requires a target.
Let's compare the clinical presentation of the two most common types of HDFN. ABO and RH.
ABO HDFN is by far the most statistically common, but usually the least clinically severe. It typically occurs when a mother is genetically Group O and her fetus is Group A or Group B. As we discussed, Group O individuals produce a unique, highly potent form of cross-reacting anti-AB that is primarily composed of IgG rather than the usual IgM. Therefore, it readily crosses the placenta.
And because it is naturally occurring, it doesn't require a sensitizing event. It can easily affect the very first pregnancy. Why is it usually mild?
Two reasons. First, the A and B antigen structures on fetal red blood cells are not fully structurally developed at birth. There are fewer targets available for the maternal antibodies to hit. Second, A and B antigens are not restricted to red cells. They are ubiquitous tissue antigens found throughout the fetal body. The maternal antibodies cross the placenta and bind to the tissues, the endothelial cells, the free-floating glycoproteins. These tissues act as a massive immunological sponge, soaking up the maternal antibodies and protecting the fetal red blood cells from concentrated, severe destruction.
RH-HDFN, driven by maternal anti-AD, is a completely different clinical beast.
RH-HDFN is highly targeted and incredibly severe. The D antigen is fully developed early in gestation, and it is located exclusively on the red blood cell membrane. There are no tissue sponges to absorb the attack. Every single antibody that crosses the placenta strikes a red blood cell.
However, RH-HDFN rarely affects the first pregnancy.
Because the mother is not naturally born with anti-D, she must be exposed.
Yeah.
During a normal, healthy first pregnancy, maternal and fetal blood circulations are strictly separated by the placental barrier. Significant mixing usually only occurs during the trauma of delivery when the placenta shears away from the uterine wall.
At that moment, a massive volume of fetal Rh-positive red cells hemorrhages into the mother's circulation.
Her immune system detects them, destroys them, and over the following weeks generates permanent memory B cells and a baseline titer of IgG anti-D. The first baby is already born and safe, but the mother is now permanently sensitized. In any subsequent pregnancy with an Rh-positive fetus, those memory cells will immediately begin pumping massive amounts of IgG anti-D across the placenta early in the second trimester, launching a devastating attack.
But this devastating disease is now almost entirely preventable thanks to one of the greatest achievements in modern immunology, RHIG, commercially known as ROGAM. The practice material heavily emphasizes the high-yield rules for administering this magic bullet. What exactly is it, and how does it work?
RAG is a manufactured, concentrated solution of passive human IgG anti-D. When we inject it into the muscle of an Rh-negative mother, we are artificially flooding her circulation with anti-D antibodies. The mechanism is essentially stealth clearance. If any stray fetal Rh-positive red cells leak across the placenta into her bloodstream, our manufactured Rh antibodies immediately seek them out, bind to them, and flag them for quiet destruction in the maternal spleen.
By destroying the fetal cells quickly and quietly, we completely hide them from the mother's own immune system. Her immune system never sees the foreign antigen, so it never learns to mount a permanent memory response. We prevent the sensitization.
The standard administration protocols are heavily tested. First, antenatal administration. We give a standard 300 microgram dose prophylactically at 28 weeks gestation. This is timed specifically to cover the slight increase in microscopic placental bleeds that occur during the massive growth of the third trimester. Second, postpartum administration. We must give another dose within 72 hours of delivering an Rh-positive baby to neutralize the massive fetal maternal hemorrhage that occurs during placental separation. Third, trauma administration. It must be administered after any clinical event that could cause fetal blood to mix with maternal blood, even if the fetal blood type is unknown. Events like a miscarriage, an ectopic pregnancy, an abortion, or blunt force abdominal trauma from a car accident.
But the exam will try to trick you with a massive logical IF. You do not just blindly give RAG to every Rh-negative woman.
The absolute contraindication. RHIG is strictly a preventative measure. It only works if the mother's immune system is naive. If her medical record shows she's already actively sensitized, if her current antibody screen is undeniably positive for a naturally formed immune anti-D administering RHIG, is completely useless. The barn door is open, the horse is bolted, her immune system already possesses the permanent memory cells. Adding passive antibody will not erase the active memory. You must document her status and monitor the fetus closely.
Let's assume she is an ideal candidate. She is Rh-negative, not sensitized, and she just delivered a beautiful, healthy Rh-positive baby. The delivery was complicated, requiring an emergency C-section. We know we need to give her RHIG, but we can't just hand her one vial and assume she's protected. The material details a mandatory two-step testing protocol, the Rosette test and the Kleihauer-Betke test.
This is about precise pharmacological dosing. A single, standard, 300 microgram vial of RHIG is designed to neutralize exactly 30 milliliters of whole fetal blood. Most normal deliveries result in a fetal maternal hemorrhage of less than 15 milliliters, so one vial is more than sufficient. But in a traumatic delivery, she may have sustained a massive bleed of 60 or 90 milliliters. If we only give one vial, the excess fetal cells will survive, sensitize her, and the prophylaxis will fail.
Step 1. The Rosette test. This is the screening test on the mother's postpartum blood.
The Rosette test is purely qualitative. It asks a simple yes or no question. Is there a significant volume of fetal Rh-positive cells floating in this maternal blood sample? We take the mother's blood, incubate it with an indicator anti-D, and then add specifically engineered indicator red cells. If fetal Rh-positive cells are present, the indicator cells bind tightly around them, forming distinct, visually obvious clusters that look like rosettes under a low-power microscope.
If the Rosette screen is entirely negative.
If it is negative, it confirms that the volume of fetal blood transferred was negligible. The technologist issues a single, standard vial of RAG, and the protocol is complete.
But if the Rosette test is glaringly positive, we have a problem. The screen tells us a massive bleed occurred, but it doesn't tell us how much. We have to switch to step 2, the Kleihauer-Betke test, to generate a precise mathematical quantification. Walk me through the chemistry of this benchwork.
The Kleihauer-Betke, or KB test, relies on a fundamental biochemical difference between adult hemoglobin and fetal hemoglobin. Fetal hemoglobin, HbF, is structurally designed to hold on to oxygen tightly, and it is chemically highly resistant to acid degradation. Adult hemoglobin, HbA, is highly sensitive to acid.
So we make a very thin smear of the mother's postpartum blood on a glass slide.
We let it dry, and then we physically submerge the slide into a harsh acid bath buffer. The acid immediately penetrates the red blood cells. The adult maternal red cells, packed with HbA, are chemically destroyed by the acid. The hemoglobin leaches out of the cells. When we apply a pink stain, the maternal cells look like pale, empty, microscopic ghosts.
But the fetal cells mixed in that smear.
The fetal cells, packed with HbS, entirely resist the acid bath. Their hemoglobin remains intact. When the stain is applied, the fetal cells turn a brilliant, dense, bright pink color.
So a technologist sits at the microscope and literally counts the number of bright pink fetal cells against a background grid of 2,000 pale maternal ghost cells.
Yes. They generate a precise percentage. If they count 20 pink fetal cells among 2,000 maternal cells, they calculate a 1% fetal bleed. We plug that 1% into a standardized mathematical formula based on the mother's total estimated blood volume to determine the exact volume of the hemorrhage in milliliters. We then divide that volume by 30 to determine precisely how many additional vials of RHIG the mother requires to achieve complete neutralizing protection. It's a grueling manual count, but it is the only way to prevent sensitization after massive trauma.
One final clinical maneuver from the practice material. What if all preventative measures failed? A pregnant woman in her second trimester has already developed a massive rising titer of a highly dangerous IgG antibody, let's say anti-Kel or anti-E. Amniocentesis confirms the fetus is antigen positive and is showing signs of severe distress. The baby is too premature to deliver safely. Is there anything the medical team can do to intervene directly with the mother's immune system?
The text highlights a drastic temporary intervention: plasmapheresis. If the maternal IgG titer is rising to lethal levels, the clinical team can connect the mother to an apheresis machine. This device continuously draws her blood out, spins it in a centrifuge to physically separate the red cells from the liquid plasma, returns the red cells to her circulation, but discards her plasma, which contains the deadly IgG antibodies.
They replace the discarded plasma with donor albumin or fresh frozen plasma?
Exactly. It is a physical, mechanical filtration of her immune system. It does not stop the memory B cells from continuing to manufacture more antibody, but it rapidly temporarily drops the circulating titer. By aggressively removing the weapon from her bloodstream, plasmapheresis reduces the volume of IgG crossing the placenta, buying the fragile fetus vital weeks of maturation time before an extremely high-risk intrauterine blood transfusion or an early surgical delivery becomes absolutely necessary.
We have covered an immense expanse of scientific territory. We have extracted the genetic blueprints, navigated the enzymatic discrepancies, traced the physical dimensions of immunoglobulins, and tracked the clinical fallout from the molecular level all the way to the trauma bay and the maternity ward.
As we conclude this deep dive, I want to leave you with a broader perspective on the discipline you are striving to master. When you are buried in these practice documents, grinding through thousands of multiple-choice questions, it is incredibly easy to get completely lost in the alphabet soup of Phaia, JKB, little c, and Delicis biflorus. It can feel like an abstract puzzle of letters and enzymes. But you must never forget what blood typing truly represents in the physical world. It is not just about mixing clear liquids in glass tubes and staring at a light box to see if they clump. It is the meticulous, critical management of a liquid organ transplant.
Because that is exactly what a packed red blood cell transfusion is. It is the transplantation of living, functioning human tissue from one biological system into another.
When you are calculating zeta potentials, when you are hunting for fading anamnestic responses, when you are hovering over a microscope executing a Kleihauer-Becker count, you are not just performing bench work. You are literally predicting, modeling, and managing how a human immune system will wage war at the microscopic level. You are the intelligence officer ensuring that the incoming tissue is recognized as an ally, not a lethal invader. As you sit down to take your ASCP board exam, remember that you aren't just memorizing pathways to pass a test. You are mastering the complex, invisible code that will keep your future patients alive.
That is the mindset you need to carry into the testing center. Go back to your trouble spots. Trust your active recall. Rely on the biological mechanisms, not just the memorized answers. You have the blueprint. Now go execute phase 1 and ace that board exam. Thank you for joining us on this deep dive into the muddy, magnificent waters of immunohematology.